Welcome to this virtual talk for the 2020 MICCAI Labels workshop with the title, Are Pathologist Labels Reproducible? The subtitle is Comparison of the 2PAC16 mitotic figure dataset with an alternative set of labels. This work was a cooperation between two pathology and two image analysis institutes from Germany and the Netherlands. My name is Christoph Bertram and I'm a veterinary pathologist. I'm going to give the introduction and discussion of this talk.
And my name is Marc Obreville. I'm a pattern recognition researcher, and I will guide you through the methods and the results section of this talk.
Mitotic figures are cells undergoing cell division that can be seen in histological images. They are very commonly counted by pathologists for tumor prognostication. It is, however, a quite difficult task, as I will explain shortly. This calls for a computer-assisted approach, and there have been quite a few challenges with the goal to find suitable methods for development of algorithms, one of which was the 2PAC 2016 challenge. The 2PAC 2016 dataset consists of numerous images of human breast cancer. Labels were created by two pathologists who screened the images independently. If candidates were only labeled by one pathologist, two further pathologists evaluated these for final decision. Problems that pathologists face for counting mitotic figures will also be reflected in datasets. Nevertheless, pathologists are the current gold standard for creating labels. First, pathologists might overlook mitotic figures. They have to screen relatively large images and find small and rare patterns within these images. Second, pathologists might misclassify candidates, a problem well known in current literature. This is because the morphological appearance of mitotic figures has some overlap to other structures, such as necrotic cells. In order to get a first impression on how much variability can be between pathologists, let's look at the published information from the AMIDA 13 challenge, which used a subset of cases from the 2PAC dataset. The two screening pathologists only annotated the same pattern in 32% of the labels. For the disagreed labels, the final decision was mitotic figure for only 31% of the candidates. The question for the study was how much difference there is between two independent labeling approaches using the same images. And we wanted to know the effects on training, measured by model scores, and the effect on testing, measured by performance evaluation.
Let me now tell you how we created the dataset. First of all, a pathologist screened all images twice for mitotic figures and for non-mitotic figures that are similarly looking cells. We then deleted the class information, be it mitotic figure or non-mitotic figure, and gave the cells to another expert, a secondary expert. The second expert then phrased an opinion on the cells, and in case of a disagreement between both experts, we had them sit together and formulate a joint consensus on each cell. Using the labels that we generated from the first expert, we then trained a machine learning solution in order to find more candidates for mitotic figures. So in case that the first expert overlooked some cells. The network was trained in order to have a high sensitivity and low specificity. The experts selected independently from those cells the ones that they perceived would be mitotic figures, which was the vast minority of cells. And then, again, in case of disagreed cells, we had the consensus done by both pathologists. Let us now compare those two datasets. The original 2PAC16 mitotic figure dataset had a mitotic figure count of 1,552. The new dataset has a mitotic figure count of 1,999. So those two datasets overlap in 1,239 cases, while 760 cases were identified as new mitotic figures. On the other hand, 246 were previously defined as mitotic figures and are now being defined as non-mitotic figures. We now aimed to assess how the consistency of the labels would be. And in order to do that, we took a RetinaNet-based model and we trained that in a cross-validation scheme. The idea was that the higher the score of the model, the higher the confidence of the model, and likely the higher the consistency of the labels is. If we do that and we then have a look comparing the model trained on the original and the new alternative set of labels, then we find that the confidence score is in general higher with the new set of labels. This is true for the agreed set of mitotic figures between both datasets, and then also this is true for the newly identified sets, or at least the majority of new mitotic figures. Now finally, we ran a couple of experiments in order to assess the performance of our new labels on the test set. We were supported, of course, here by our co-authors who are in the possession of the labels of the 2PAC16 test set. Now, when we do this and we train the same model pipeline, one time using the original 2PAC16 labels and then using the alternative set of new labels that we provide and published here on this workshop, then we can see that it made quite a difference. The original set led us to an F1 score of 0.549, while the new set had a F1 score of 0.735. So this is, from our point of view, quite a remarkable difference in the F1 score.
Let's discuss our results. Our results indicate that pathologist-defined labels are not very reproducible. We conclude that labeling should be done with highest possible degree of diligence in order to improve consistency and completeness of labels, as this obviously influences training and test performance. We also highlight that comparison between datasets should be done with caution. For example, labels of open access datasets were created by different pathologists and different methods. If, however, a study aims to use the same deep learning method and compare results between open access datasets, the methods might work better or worse on one dataset only due to the labeling process, as seen in our work. Or, if studies want to transfer the algorithms to other datasets, with or without model optimization, results might vary just because of the differences in the labeling process, which is also obvious from our result. In this image, we highlight the difference between the labeling processes between the original dataset and the alternative dataset. For both approaches, manual screening of the images by pathologists was the foundation of labeling. However, for the original dataset, the images were screened independently by two pathologists, and for the alternative dataset, one pathologist screened the images twice. For the later approach, the first pathologist also labeled similar patterns, and labels were blindly reviewed by a second pathologist, which reduced the time investment for the second pathologist. The labeling method of the alternative dataset basically followed the paradigm of Viola and Jones, of having an initially highly sensitive detection, followed by a secondary classification with high specificity, achieved through a dual expert consensus. We also added an algorithmic augmented labeling approach that was aimed to detect miscandidates with high sensitivity. Again, high specificity was achieved by pathologist review of all potential candidates. On this last slide, we compare different methods that can be used to generate labels. The gold standard are pathologists that are highly trained experts. However, they have inter- and intra-observer variability and are prone to fatigue of concentration. Increasing label quality can be done by repeated screening and multi-experts, which will, however, significantly increase time investment. We can also use algorithms to generate labels. Algorithmic augmented labeling requires decision by annotators, and fully algorithmic labels are not reviewed by experts. Detection of misannotations aim to identify additional labels that were missed by screening of pathologists. This approach should have a low algorithmic detection threshold in order to detect all candidates and to generate many false labels that can reduce confirmation bias of pathologists. Algorithmic pre-annotation of slides aim to increase dataset quantity, and overall good algorithmic performance is required. Labels are ideally reviewed by experts, and our research group is presenting a study on this labeling approach at MICCAI 2020. We have determined that speed of labeling is significantly increased, but algorithmic label errors are commonly overlooked. Fully algorithmic labeling is the method most prone to labeling errors and is only acceptable for training data. While there are different labeling approaches, we highly encourage future research to further evaluate benefits and limitations of these different labeling approaches. Thank you very much for your attention.